days of the endless summer. These are the days when time is now. There is no past. There's only future. There's only here. There's only now. Okay, and that was Van Morrison. These are the days. I figured we might as well stay with the theme since this is the second half of Neurons Part 2. And why don't we begin? So we're in the first half of the lecture. We talked about Ramon Cajal. We talked about neurons as cells and the discovery that they were discrete units. We talked about the structure of various neurons and the structure of the synapse. Right, it's a gap in presynaptic membrane, postsynaptic membrane. We talked about the types of cells in the brain, the glia, the oligodendroglia, the astrocytes, and all that kind of stuff. In this lecture, we're going to talk about the communication process within neurons and between neurons, right? And remember, when we combine that all together, that's neurotransmission, yeah? And that's what everything is based on. That's how we experience our world, is through the function of these tiny little discrete units, yeah? So we're going to be talking about a little bit about resting membrane potential and the action potential and the electrical impulse. So let's take a look at a membrane. Okay, so here we have your membrane, which is also called a phospholipid bilayer. And why is that? Well, each discrete unit here, the head and the tail, is called a phospholipid. Yeah, and so you'll notice that all the heads are in one area. They're all bound together in one area, and then the tails are all on the inside, right? So the reason for that is because there's water on the inside and outside of the cell. And the heads of the phospholipids are hydrophilic, which means they're comfortable in water. They are attracted by water, right? So they face out. The tails of the phospholipids are hydrophobic, which means they don't like water. They want to avoid water, so they all face in, right? And now this then results in a really nice sort of bilayer that is what comprises the cell membrane. Yeah, so every neuron, the lining of it is a phospholipid bilayer. Yeah. Now this is a very simple representation. Yeah, in the next slide, we're going to see that this membrane has a whole bunch of stuff in it, and the reason for that is because I want to make sure that you're clear when, for example, receptors, those are in this membrane. Yeah, they're in the membrane of the postsynaptic dendrite. Yeah. So there are also ion channels that are embedded in this membrane. Yeah. What we're talking about right now is called the resting membrane potential, which is negative 70 millivolts, right? That's millionths of a volt. And why do we care about that? That's basically an electrical charge. Yeah. So our membrane, the cell membrane of a neuron, has a negative 70 millivolt resting charge. Yeah. And why does that matter? Because what happens when neurotransmitters bind with receptors is they change that potential. Yeah. And so if they increase the millivolts, that's going to produce an excitation. If it decreases, then that's going to produce an inhibition or a reduction. Yeah. So let's take a look at another image of a membrane, of a cell membrane. Here we go. So you can see here, again, if you look on the right-hand side of the image, the hydrophilic heads and the white parts are the tails, right? So they form a membrane, they form a layer. And inside that layer are embedded all of these different proteins. Yeah, there's alpha helix protein. There's you got your glycolipids, your glycoproteins, all that kind of stuff. Including on the far left-hand side of the image is a protein channel. That's an ion channel. Yeah, it's made up of protein, right? But what it does is it sits there closed until it gets activated, and then it opens up and it allows ions to come into the cell. Yeah. Okay, so let's talk about that resting membrane potential. And how do we know about this resting membrane potential? Well, it comes to us from a couple of gentlemen named Hodgkin and Huxley. Yeah, And these guys here won the Nobel Prize in 1963. They did it. They started in the 1930s. Yeah, that's when they began the experiments that earned them a Nobel Prize in physiology in 1963. And what they were doing was they were working with giant squids. Yeah, and the reason for that is because the giant squids have a very large axons. Yeah, or large neurons altogether, right? So what they did was they did experiments. They wanted to see a flow of electrical current through the axon membrane. Yeah, they wanted to identify how is an electrical charge sent from one part of this cell to the other part. Yeah, and so they worked with the giant squid axons because they were easier to see, right? Remember, this is the 1930s. We don't have, Google and sophisticated technology. They're working with microscopes and that sort of thing, but it's really rudimentary compared to the things we have now. Yeah. So, but the axons of the giant squid were large enough so that they were able to observe and manipulate the action potential, and that's where the idea of the action potential comes, and that's why we thank Hodgkin and Huxley for that finding. Okay, and so here we go. Action potential is that electrical current that travels down the axon. Yeah. Now, some important things to note about the action potential are that it is all or nothing. Yeah. And what that means is it either fires or it doesn't. You don't have different voltages of the action potential. It doesn't, you can't turn up the volume or turn the volume down. It either fires or it doesn't, and it's the exact same pulse each time it fires. Yeah. So it's all or nothing. It will either fire or it won't fire. If it does fire, it fires in the exact same way that it did before. Yeah. The way that we get to an action potential, we need what's called depolarization past the threshold of excitation. And all that really matters is a change in the membrane potential from about negative 70 millivolts to about negative 50 millivolts. Yeah, that's all it takes, a real tiny change in that electrical charge. Yeah. And when that happens, we hit what's called the threshold of excitation, which we'll look at in another slide. But that's once that action potential fires, then typically what it results in is the release of neurotransmitters at the end of the axon in the terminal buttons. Yeah. Okay, so we talked about how it's initiated. It's initiated by a change in that resting membrane potential. How does that resting membrane potential change? Well, we have two different kinds of postsynaptic potentials. We have inhibitory postsynaptic potentials and excitatory postsynaptic potentials. What matters here is basically these are messages that are being sent. Yeah. And one is telling the neuron not to fire. That's inhibitory postsynaptic potential. And the other is telling the neuron to fire. That's the excitatory postsynaptic potential. Yeah. So by binding with receptors on the dendrite, yeah, these molecules can cause either an ISP or an ESP to be started. And it's not ISPs and ESPs are happening all the time. Yeah. So it's not just one inhibitory postsynaptic potential that causes the neuron not to fire, right? There's what's called summation. There's summation across space, which means the distance across the neuron membrane, right? And that an inhibitory postsynaptic potential in one area and another in the other, they build up together, right? They get added together. Same thing goes with excitatory postsynaptic potentials, right? There's also a summation across time. So if an excitatory postsynaptic potential is caused, yeah, it can. If there are others being caused around the same time, it can all be added together or summed, right? So there's a summation across time. Enough of those excitatory postsynaptic potentials occur, and you have then an action potential. If enough of the inhibitory postsynaptic potentials occur, you're going to have a lack of an action potential. It's not going to fire. Yeah. Okay, so again, I don't want folks to get too worried about all of the ins and outs about the negative 70 millivolts and the 50 millivolts and all that other stuff. But what matters is that neurons have a resting membrane potential, right? And that's at negative 70 millivolts. That's what we see here on the left-hand side. That's where all the lines are starting except for that dotted line, right? And the reason for that is because the dotted line represents the threshold of excitation. That means the level of electrical charge that the neuron membrane has to reach in order for an action potential to be generated, right? So it's getting both inhibitory postsynaptic potentials, which are reducing its chance of fire. That's what the blue line is, and you see how it goes down rather than up. And it's also getting excitatory postsynaptic potentials. That's the yellow line that's going up, right? So enough of those excitatory postsynaptic potentials come along, and they will raise the membrane, the resting membrane potential to the threshold of excitation and cause then that firing. And you got your sodium ions just rushing in along the action or along the axon. Yeah. So it's that change in the neuron potential. Yeah. It doesn't really matter. I'm not going to ask you about sodium channels. I'm not going to ask you about electrostatic pressure and diffusion and that sort of thing, right? But know that it's called an electrical, it's an electrical pulse because of these various ions and what they're doing, right? We have sodium, potassium, that sort of thing. So what's actually happening is we're seeing sodium rushing into the axon. That's that charge that runs down there. Yeah. And again, I don't. What I care about is that you understand that there's an electrical charge that runs down the axon. Yeah. You don't have to know about sodium and that sort of thing until pre-med. Okay, and so here's a nice little GIF of the difference between the action potential in myelinated axons and non-myelinated axons. Yeah. So on the left, non-myelinated. On the right, myelinated axon. Right. And what you're seeing is, the charge starts up at the body, up at the soma, and it's already down to the dendrites on the myelinated axon before it's even halfway down the axon of the non-myelinated neuron. Yeah. So myelin speeds the process. How does it do that? Because basically the entire myelinated area gets activated from the action potential, and so it's almost instantaneous. And all that has to happen is an action potential at each node of Ranvier. Yeah. So rather than the action potential slowly traveling down the axon, it just goes beep 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 beep, one to the next, right? From one node of Ranvier to the next. That process is called saltatory conduction. As far as I'm concerned, it should all be called saltatory conduction because they're talking about sodium ions in general, right? But remember, though, saltatory conduction is what happens when an axon has a myelin sheath, and the action potential is traveling down that axon. It's jumping from one node of Ranvier to the next, and that's the definition of saltatory conduction. And it's rapid transmission of the action potential, right, as you see. Okay, so that's pretty much all I want to say about communication within neurons. Let's get on to communication between neurons. And it's important, again, to stress that now we're going to be talking about a chemical process, whereas previously we were talking about an electrical process, right? Remember, neurotransmission is an electrochemical process when you put it all together, right? But we're breaking it down so that we can understand the various aspects of it. So here we have an image of a schematic, basically, of a neurotransmitter, right? And so neurotransmitter on the top, receptor on the bottom. And you can see very specific shapes on that neurotransmitter and very specific areas on the receptor that correspond to those shapes on the neurotransmitter. Yeah. So this is what happens. This is the idea of neurotransmitter release when it hits that presynaptic terminal button. The neurotransmitter, and it can be acetylcholine, it can be glutamate, it can be GABA, it can be dopamine, serotonin, is released from that neuron, and it floats across the synapse, and it will bind in a lock and key fashion, right? Now we have two different kinds of receptors that we're going to worry about. There are more than two different kinds, but let's just focus on, again, this level. So ionotropic and metabotropic receptors, and what that basically refers to is ion channels and G protein receptors. Yeah. So the ion channel is a fast acting receptor, and metabotropic is a slower acting receptor. Yeah. So, but either way, when the neurotransmitter binds with its receptors, it can result in either an inhibitory postsynaptic potential or an excitatory postsynaptic potential. And so here we have a nice little image of both an ionotropic receptor and a metabotropic receptor. Ionotropic receptor is also called an ion channel. Yeah, and so on the left-hand side, we see these green neurotransmitters binding with the ionotropic receptor with the ion channel and causing it to open up and allow for ions to flow inside of the neuron. Yeah. On the right-hand side, we see this metabotropic receptor, which is also called a G protein coupled receptor. And the reason for that is because if you look down at the bottom there, those blue sort of oval things, those are called G proteins, and basically they are attached to this receptor, and then they get sent. Once the receptor gets activated, they get sent to other areas in the cell and activate other processes, right? One of the things that it can do is open an ion channel. Yeah. But it also does a lot of other much more complicated sort of cascading things that can change, like the way that RNA is transcripted. It can change all kinds of things in the cell. So remember, though, that the G protein coupled receptor is slower. The ion channel receptor is faster. Yeah. Okay, so your textbook talks a little bit about glutamate and GABA and that sort of thing, right? What matters for you guys is that glutamate is an excitatory neurotransmitter. When glutamate binds with the receptor, it is only going to produce an excitatory postsynaptic potential. Okay, it's not going to produce an inhibitory postsynaptic potential because that's not what the lock and key system does with glutamate. Yeah. Now. So glutamate, excitatory, right? And then we have GABA, and that's inhibitory. And when GABA binds with the receptor, it's likely to produce inhibitory postsynaptic potentials. Yeah. And of course, again, we have a whole bunch of different kinds of neurotransmitters, right? Who can produce various different things. But if you want to talk about an excitatory neurotransmitter, glutamate is the number one. You want to talk about an inhibitory transmitter, one that slows things down, calms, reduces the firing in a neuron, that sort of thing. That's going to be GABA. Yeah. All right, and wanted to give you guys a good image of exocytosis. Yeah. Exocytosis is the release of neurotransmitters into the synapse, right? So we're going to follow these arrows starting at the top of the image with the endosome, right? The endosome is where vesicles are made and where they're packaged with neurotransmitters. Yeah. So from the endosome, it does this process of separating off a little circle of itself, and that becomes a vesicle, right? Each vesicle has a cluster of proteins in its membrane. That's what those red ovals are on the left-hand side of your screen there. Yeah, each vesicle has these proteins. These proteins can bind with proteins that are in the membrane of the presynaptic terminal. Yeah, and that's what that exocytosis is. That's the beginning there. These proteins bind. See, the red proteins are binding with the yellow proteins. Yeah. And once they've bound, they're basically forming a what's the word? Well, it's kind of like a zipper process, yeah. So as you can see, those proteins inside the membrane will then open up, right? They will spread apart. When those proteins spread apart, as you can see, the membrane of the synaptic vesicle is fused with the axon terminal membrane, right? And so that's how it releases the neurotransmitters without opening up, any sort of area where bad stuff can come into the cell. Yeah. So it's an amazing process because it's basically, becoming part of the cell membrane, releasing its molecules, and then closing back up. As you can see on the right hand side of the image, it closes back up and disconnects from the membrane, and that's endocytosis, which we don't worry so much about. And then it goes back up to get recycled, to get refilled with neurotransmitter, and to do it all over again. Yeah. So it's a basic sort of process, but it's also amazingly eloquent in that it solves, one of the major problems is how do we get, molecules from inside the cell to the outside of the cell without exposing the cell to any kinds of damage or waste or negative things coming inside. Yeah. All right, and then I know that this is a very busy picture here, but I wanted to use this picture because it really gives you the whole enchilada, yeah, all together, right? From where the vesicles are synthesized, they are transported, they get hit by an action potential, they release their neurotransmitters into the synapse. Neurotransmitter molecules go across the synapse, they bind with the receptors that cause that postsynaptic activity. We talked about all that. So then what happens? Well, then what happens is we have to terminate this process, right? We can't allow the vesicles to continue to bind and to continue to release neurotransmitter. Yeah. Just like you need something that's going to stop your car. You need something that's going to stop the neurotransmission, right? And so we have two different ways of doing that. We have enzymes and we have reuptake. Yeah. So you can see number 7 here, we're kind of going clockwise, and I'm on number 7. It says reuptake of molecules by transporter proteins. So there are proteins inside that presynaptic membrane that the neurotransmitter molecules combine with, and they get brought back in. Yeah. And once they're brought into the presynaptic membrane, they can be broken down, like these enzymes. MAO, monoamine oxidase, is one of the major enzymes for breaking down neurotransmitters. That's why you may have heard of monoamine oxidase inhibitors. Those are used for various mental illnesses and such, right? And the reason for that is because it allows for more of the neurotransmitter to remain in the synapse if you inhibit this enzyme. Yeah. So we have this reuptake, and it can pull neurotransmitters back into the presynaptic membrane, where they then get broken down and then get those pieces get put back together into neurotransmitters again and put into the vesicles, and the whole process goes all over again. But enzymes also exist in the synapse themselves. Yeah. So we can have enzymes that the neurotransmitter, if it doesn't get brought back in through reuptake, it can also be broken down in the synapse itself. Yeah. And so one of those two ways, yeah, enzymes and reuptake. Okay, and then this image is of an acetylcholine synapse, and the reason we're using this is because I think it's very straightforward and it gives you a easy to follow sort of image, I think. So acetylcholine is a neurotransmitter that we need for things like attention. Yeah. The more active our acetylcholine systems are, the better we're able to maintain our attention on something. Yeah. It also helps us with the speed of processing of information. Yeah. That's one of the reasons why people like smoking cigarettes. And now, of course, it's vaping. Yeah. But the main action of nicotine is to enhance the action of acetylcholine in your brain. Yeah. So that's basically it. You can get a buzz from a cigarette the first couple of times you smoke or whatever, but you develop a rapid tolerance to that. But in general, it's not really, you don't really feel much when you smoke tobacco mentally, right? But it does enhance your ability to focus. It enhances your ability to maintain your attention, yeah, and those sorts of things. So acetylcholine is a very important neurotransmitter. It's one that a lot of people are familiar with, or manipulate themselves through vaping of nicotine and nicotine gum and all that other stuff. Yeah. Also, why when I was teaching in-person classes and we had an exam or that sort of thing, I'd always tell my students who smoke to make sure that they don't come to class for a quiz or whatever jonesing for a cigarette, because they're going to have lower levels of acetylcholine. They're going to struggle in their exam. Yeah. Okay, so what matters is that we have a process by which these acetylcholine molecules are made, right, through an enzyme, right? And so we have these two parts. We have an acetyl molecule and we have a choline molecule, right? So that's the green one, acetyl, and then the pink one is choline, and they get put together by an enzyme into acetylcholine, and then they get packaged in synaptic vesicles, right? Now, once that gets released into the neurotransmitter, or I'm sorry, into the synaptic gap, and it binds with the receptor, and it has its effect. Then it releases from that receptor, but does not get brought up as a whole. Yeah. Instead, like I said in the previous slide, this one gets hit by an enzyme called acetylcholine esterase. Yeah, ACHE. That's down here. It's marked number 2, and it gets metabolized in the synapse itself into choline and acetyl, and then those two precursor pieces are what get brought back up into the synapse. Yeah. So we can have reuptake where the whole molecule gets sucked back into the presynaptic membrane, or we can have this enzymatic degradation process that can happen in the synapse itself. Yeah. Okay, and that'll do it for chapter 1 for neurons and such. I talked a lot about this in the first lecture, so just have this slide here just to remind you. Don't forget about your discussion. Don't forget too that next week we're going to be doing double work. We're going to have twice as many chapters to read. You're going to have your first paper to do and that sort of thing. So you don't want to let that slip. All right, so for the discussion, remember you need to read the article. Don't forget your quiz is on this Friday. And then next week, we're going to do chapter 2 and 3. We're going to have a quiz on chapters 2 and 3. We're going to have our second discussion and your first reaction paper on The Brain with David Eagleman. This is, I think, a fascinating film where he's talking about what reality is and what our brains perceive and how we sort of compose reality for ourselves, right? I hope you enjoy the film. I want to thank you very much for hanging out with me throughout this entire lecture. If you have any questions or concerns, please contact me. I'm more than happy to help you. I want to work with you. Until lecture 2 and chapter 2, hui ho.